Well, thank you, David, for that kind introduction, and it really is a pleasure to be here. Our collaborations with Yale really have been fun and exciting, and kind of seeing where we can take these in the future is really gratifying to see it. This is, in presenting this specific talk, this is sort of the, bringing coals to Newcastle thing, or perhaps in this case I was thinking it's sort of bringing pizza to New Haven, where, this is really where a lot of the initial and really most sophisticated research on alcohol and HIV has been conducted. So we're really early on in this process. I want to acknowledge collaborators at Brown and also here, and the funding support from NIAAA with a number of grants, and also from the Brown CFAR has been also helping us bring our alcohol and HIV worlds together at Brown. I also do need to acknowledge the participants in the SUN study and the SUN investigators, as I'll be presenting data from that particular thing, so you probably won't have time to read all that. So the Alcohol Research Center on HIV has a number of research components. I'll quickly go over those before I kind of dive into sort of some more recent data. One of our projects looks at alcohol and HIV-associated brain dysfunction. It's a neuroimaging study, longitudinal. We have a brief alcohol intervention for HIV-infected MSM in a primary care setting, which I'll talk about today. We have a similar type of intervention that's a dual focus on alcohol use and sexual risk behaviors in the emergency department that Peter Monti is the PI of. A recent sexual minorities research resource core that Don Operario is the PI of, and we're just starting to kind of explore the connections with Yale, and I think those will be quite productive. And then also a mechanisms of behavior change resource core that was funded this past fall. The sort of theme of all of those is that we're really in sort of our initial steps here, and David was kind enough to call me a generalist or a master of none, perhaps, but we're sort of new to this work, but we bring a large set of alcohol-related research and methods that we hope will be useful as we go further. So the kind of key theme of our center, and this is a slide that's existed since the site visit a number of years ago, is that alcohol sits in the middle of a complex nexus of biological and behavioral factors that impact the depth and the breadth of the HIV epidemic. So we can see that alcohol use affects sexual risk behaviors, which of course affects transmission of the virus, perhaps increases susceptibility to acquiring the virus through a variety of mechanisms, sort of not as well understood. Alcohol certainly has effects on medication adherence for people who are infected, perhaps interacts with medications and their effectiveness. Again, that's not clear. Alcohol and antiretroviral medications can impact the liver or are certainly processed there. And then liver dysfunction, alcohol use, and HIV itself all can have an impact on the brain, which of course then goes back and affects the behavioral aspects of alcohol use, medication adherence, and sexual risk behavior. So we're trying to kind of elucidate a little bit of each of those pathways to understand what are the different mechanisms involved and how do we make an impact in that. So just very briefly from a meta-analysis, just to set the stage in case some of you do not have sort of alcohol and HIV as a central focus of what you do. Alcohol consumption, the risk of incident HIV infection is a significant association, more than double the odds of binge drinkers getting an incident infection versus non-drinkers. That's from a meta-analysis. A recent study in MSM found that binge drinkers were 60% more likely to have an incident HIV infection compared to non-drinkers. So certainly some strong associations, which means that the people who get infected are more likely to be heavy drinkers. Once infected, the odds of unprotected sex among people living with HIV is also a significant association, where problematic drinking is associated with a 70% increase in unprotected sex. And then on the other side of that, of concern both for the individual's health and of course for transmission, is that the odds of being completely adherent to medication or optimally adherent reduce with any drinking and reduce with at-risk drinking. And that's a very strong, consistent association that's been seen in men and women, intravenous drug users, numerous countries. This is one of the most consistent associations, I would say, that we see. Scott Braithwaite and colleagues here looked at it's not just that binge drinkers are different as people, so that they're always more non-adherent. It seems that the non-adherence is most strongly associated with the actual consumption of alcohol. So a potential causal association. But even with that causal association, there's still, what are the reasons for that non-adherence? It could be forgetfulness, or it could be conscious decisions. And Seth Kalishman's recent work suggests that people who believe there is some interactive toxicity between alcohol and antiretroviral medications are less likely to take medications on days they drink, and that about a quarter of drinkers in his samples report those types of beliefs. There's, of course, also general disruption associated with drinking problems, decreased healthcare utilization. And I think sort of intriguingly that a study looking at provider interaction with patients found that heavy drinkers tended to have worse interactions with their providers. Their visits were shorter, and there were less engaging counseling statements. So they were a little more surface level visits. So it could be that they don't talk about adherence and some of the problems with adherence as deeply as they might with non-hazardous drinkers. Of course, there's strong associations also with alcohol, serious illness, and death. Non-AIDS events among HIV-infected adults who drink are higher. Death is associated, a higher death risk for women drinking more than 7 drinks a week. History of heavy versus low alcohol consumption also associated. Although a recent study found no association between heavy drinking and death when controlling for viral load and Karnofsky score, which does suggest that it may in fact be viral load and sort of general medical health problems caused by drinking that are contributing to that death. So although this sort of sounds like a negative study, I think it actually suggests that maybe that's one of the mechanisms, as opposed to other causes of death, such as violence and accidents and so on, that could be associated with drinking. There have been a lot of studies looking at alcohol and HIV outcomes, but I would say the results are relatively variable. But just to give you a sense of that, Azar reviewed some of those studies from the early part of the decade. 5 found a positive association with greater drinking and increased viral load. 4 found associations with decreased CD4 count. And the 3 that didn't find associations seemed to have some possible limitations in their sample or the methods used. Chander's study suggests that the odds of being adherent down there, the adherence is particularly low, so the odds of being adherent is reduced with hazardous drinking. Virologic suppression is also reduced, although you see that association is not quite as robust. It almost crosses 1. But within that study, it was also the case that it was not just drug use. It wasn't sort of that multi-morbid conditions, that the association happened whether or not there was drug use present or not in the individuals. So pretty strong effect. Just a couple of more recent studies, the odds of unsuppressed viremia in a sample with fairly high rates of lack of suppression, a lot higher in at-risk drinkers. One recent study found no association, found an association with drinking and viral load, but only among those on heart and not those not on heart. So again, whether or not people are taking their medications, I would suggest it probably does go potentially through adherence if there's that association. That one did not find an effect on CD4. So I think in some cases you see effects and other times you don't. Again, this just sort of illustrates that the risk of having a decline in CD4 counts, again, higher in people with frequent alcohol use compared to people who are moderate drinkers, and especially elevated in people who use alcohol and crack. But you see most of the risk seems to come with alcohol itself, is where we see sort of a lot of that hazard ratio being due to just alcohol use. So, those are sort of the key HIV outcomes that you see the most on, viral load and CD4. And of course, Amy Justice and colleagues here have developed a YACS index as a way to measure some of the other biomarkers that might be related to the odds of a patient surviving. And it incorporates information on age, hepatitis status, viral load, CD4, but also liver function from the FIB4, kidney function, and hemoglobin as other predictors. And that one, generally, if you follow this trend here, does show, if you go up in the grayscale there, a steeper line associated with quintile of risk from the VAX score compared to an index that doesn't have one, so it being a more robust predictor of the odds of death. So what we don't know, though, is really what about some of those components and how do those relate to alcohol use, and can we tease that apart? So that'll be what I'll talk about now, some of those components, and then provide some data on that, and then go into talking a bit about interventions as well. So in terms of the liver disease has surpassed AIDS defines illness as the cause of death in people living with HIV. It was present in about 50% of deaths from liver disease in HIV-infected patients. It's certainly well known to be hepatotoxic in high doses. And there are some hypotheses about that HIV infection may exacerbate these effects. And a lot of mechanisms have been suggested: increased gut permeability, increased inflammation, fat accumulation, insulin resistance, potentially an interaction with HIV medications. I don't really think that the data are very solid on whether or not they're sort of like biologically plausible, but I don't think we really have the causal data to suggest whether or not that's actually happening. We do know that within HIV patients that alcohol abuse and dependence is associated with increased fibrosis and cirrhosis, as one would expect. That's also been shown in people with co-infection. Greater AST to platelet ratio index as a biomarker has been associated with hazardous drinking. But there's still questions about whether those have been the best markers used to date. In a recent study, hazardous alcohol use was only found to be associated with higher AST and ALT in people who were co-infected with HCV. So again, the moderators of these associations are not clear. In mono-infected, they did not find an association. I think that could also be a sample size issue, but it didn't seem to even be a trend. So a lot of the studies so far have reported AST and ALT. FIB4 is an alternative index of liver function that kind of harnesses measures, both of those measures, and also platelets. And it's been found to be the best predictor of cirrhosis and fibrosis in co-infected persons. And it has been shown to be elevated even in mono-infection. So there may be some direct effect of HIV itself on the liver, or it could be the medications. The higher viral loads are associated with FIB4, so at least we do have some data on that. It does predict hepatocellular carcinoma in HIV patients. The only study, though, that I found that showed the direct association over time between alcohol use and infected and uninfected individuals found no association between alcohol use and FIB4. So that was a bit surprising. It was only in women. And so, again, that sort of suggests that we need to look a little more deeply there about that association. So two of the other components of the VACS are hemoglobin and EGFR. And those really have been, in general, there's not a lot of work on those done in alcohol use in general, and certainly not in HIV-infected patients. We haven't seen those parameters yet published. So with hemoglobin, alcohol is actually associated with moderately higher values, so it potentially has a positive effect, although what the meaning of it is biologically, I'm not sure we know. It's been associated with reductions in red cell count, but increases in immune corpuscular volume, so it could be that there's something more complex going on there than just sort of a raw measure of hemoglobin. We see similar effects with smoking, and there's been hypotheses that that's related to COPD, and as people have more breathing problems, perhaps hemoglobin changes in some way to compensate. But I did not find studies published in HIV patients, although I know that sort of the data are here as one place. For kidney function, rats fed a high percentage of calories from alcohol do end up showing reduced GFR and increased catalase activity. But in humans, we actually find sort of the opposite in observational studies, and evidence of lower risk of reduced GFR with consumption of 7+ drinks a week. And also that drinking frequency and quantity are associated with a lower risk of chronic kidney disease. So it's possible, again, alcohol has some potential beneficial effects, and we really need to know within people infected with HIV, does it have any of these beneficial effects, and ultimately what might be the meaning of those down the road. When we think about the VACS index as one of the best predictors of survival, we really want to know those components, and it could be that people are going up on one component and down in another, which would give us sort of a muddied picture. So that's one of the key questions here. How does alcohol relate to each of those components? And then within those, you see a lot of studies tend to make a rough cut. Either they're using the audit or a measure of alcohol abuse or dependence. But we don't really know what drinking parameters are the most important. Is it how often you drink? Is it how many drinks total you're putting through your body and your system? Is it heavy drinking itself that's important? So what kind of classifications are the most relevant? In the interest of time, I will kind of shortcut that discussion and tell you that going in, we thought that frequency of heavy drinking would be probably the most important in that, and hazardous drinking kind of correlates with that so heavily that they're often very difficult to separate. But we thought those would generally be the strongest, and that's generally what we find. In all these data, we did look at drinks per week and drinking frequency, and we just don't see the associations as robustly. So heavy drinking seems to be sort of the best marker in that. And then we don't know within that, sort of importantly, mechanistically, if we see an association between alcohol use and worse HIV outcomes, is that pathway going through medication adherence, or are there other pathways that alcohol is having direct effects? And likewise, does the effect of alcohol vary by heart status, so that people who are taking antiretrovirals perhaps have worse liver function if they're also drinking heavily because they're putting a combined stress on the liver, or perhaps being virologically suppressed might protect your liver in some way. So I think there's a lot of questions there as well. And we don't know about co-infection as well. We would certainly think that HCV could enhance some of these associations. Certainly we think of liver function. People with hepatitis of any sort, we would expect that there would be a stronger association between drinking and alcohol use and liver function. So the method was the SUN study. This is called the Study to Understand the Natural History of HIV and AIDS in the Era of Effective Therapy. This is a CDC-supported longitudinal cohort study. The N that we have sort of valid data for at baseline and going forward is 689. Individuals in this cohort were only exposed to combination highly active antiretroviral therapy. So these were not people who were on single therapies, monotherapies, and have been switched later. So these are people only initiating sort of the modern era of therapy. They were followed for 7 years. There's a couple follow-ups still being finished up, but I cut it at 7 years. They have assessments every 6 months, and those include kind of the typical serum assessments that you would get, and then the behavioral assessments as well. In total, that gives us 6,754 assessments, a mean of almost 10 visits out of a maximum of 14, or sorry, out of 15, because the baseline visit counts as sort of day 0. So that's sort of the sample. They come from these sites. There were 4 clinics, Minneapolis, Providence, St. Louis, and Denver. These are relatively generalist clinics, so these are not sort of a VA or any kind of specific population. So in that way, you do get a fairly good representation of men and women relatively, although the skew is much more towards men. To describe the demographics of the sample a little bit, it is only just under 25% female. MSM are the largest group in this at 61%. There's generally, for an HIV sample, this might be on a somewhat higher educated level than I think a lot of people might find. Only 12% were below a high school education. Reasonably diverse sample, almost 30% African American, about 10% Hispanic Latino. 12.7% HCV co-infection, so it's certainly there, but not sort of the dominant mode of is to be co-infected. And then alcohol use at baseline. So there were 31.6% were non-drinkers at baseline. Now, of course, people transition out of these categories over time, and I use sort of alcohol use as a time-varying covariate in this. But just to give you a sense, it's easier to look at it at one time point. 39.2% had drinking without heavy drinking. So there's really quite a large group in that. In these analyses, we did initially sort of do these splits, but we did find that the drinkers in that middle group who were drinking without heavy drinking, we didn't see a lot of differences on really any measure that we were looking at. So for this analysis, I'll collapse. What I'll be doing actually is a contrast code where I compare heavy drinkers versus these two groups, and then compare within these groups, does frequent heavy drinking make a difference relative to infrequent heavy drinking? So if you can see sort of a dose-response association, gives you a little stronger ability to draw some inferences about what alcohol use itself might be doing. So this is the analysis plan. We used a repeated measures analysis using generalized estimating equations. HCV and demographics were sort of measured at baseline. Alcohol use was a time-varying covariate within that. So at each time point where we analyze it, we put in the alcohol use value at that particular time point, and then we're predicting the particular biomarker at that given time point. Adherence then was also used as a time-varying covariate. So from that same time point, we then test the interaction between hepatitis C and time-varying alcohol use, and the interaction between time-varying medication status and alcohol use, that individuals may go on and off of medication during this trial as well, or not trial, during this observation. So to start with adherence, this is sort of where we expected to see the strongest signal. Across visits, 18% of valid patient observations included at least one missed dose in the past 3 days. Now this, of course, is a pretty rough measure in a lot of ways. One that it just only has 3 days, and of course it's only self-report. So that's important to note. So we kind of see this as people who are probably pretty non-adherent in general. If they're willing to report that they didn't take it in the last 3 days, my guess is there's something going on. These showed pretty strong associations with heavy drinking. So the odds ratio for heavy versus non-heavy drinking, if we just kind of make that kind of broad cut, 70% increased risk of non-adherence. So that's actually pretty consistent with the meta-analysis data. So we see it sort of in the same sample. If we look at frequent heavy versus infrequent heavy, we get a 50% increase. So it does matter how often you're drinking heavily. So I think that's important too. It's not necessarily just the people. This is controlling for a number of potential confounds. It looks like frequent heavy drinking is worse than infrequent heavy. So that one is pretty straightforward. All the others had some sort of complexity in it. And talking to Amy and David today, I think that seems to be sort of the law of the land, that adherence we see really pretty straightforward associations. When we then have to think that the pathway goes through adherence to some other outcomes, so many other clinical and psychosocial factors can be interfering in that association. So we looked at log viral load. We had about just under 30% of valid patient viral loads were in the detectable range. There was a weak, non-significant association with heavy drinking. And so it's sort of almost a trend. It's not a particularly robust effect. And importantly, frequent heavy versus infrequent heavy drinking actually would have gone in the wrong direction. We would expect this to be a positive association. So really, in viral load, we see a lot of complexity. And that could be a lot of, we don't have sort of reverse causation. We need to think about some of those things. People who get more sick may be drinking less, and that may be the times at which your viral load is high. Sort of a lot of different factors going on. But for all those reasons, we see sort of a relatively muddy picture. And again, that's consistent that about half of studies that look at this find an association and half don't. It was not significantly moderated by HIV med status. So if we take people only on antiretrovirals, we did not find any difference there either. CD4 count was different, though. Across visits, CD4 cell counts were reasonably high in a good range. There was a trend towards a negative association with heavy drinking in that. So some heavy drinking compared to no heavy drinking, we had a P of 0.05. When we had frequent heavy versus infrequent heavy, however, again, we had a negative coefficient there. Again, we would expect a positive coefficient. So, missing that. That's actually in the right direction, but it's important to look at the confidence interval there. So we expected a dose effect. The sign is in the right direction, but there really didn't look to be much of a dose effect. But there was a significant interaction with HIV meds. So this is in contrast to one of the studies I reviewed, which is why I went through that kind of comprehensive list, that we found a significant interaction on CD4 count specifically. So among those on HIV medications, we see a reasonably robust association of reduction of an average of 20 points on the CD4. Again, though, we do not see strong evidence for sort of a dose-response association there. So something about heavy drinking was associated with reduced CD4 count. It was robust to control for medication adherence in a cross-sectional analysis. And I'll talk about causal modeling in a minute. And there, the picture is a little more complex. So FIB4, I would say actually FIB4 is in some ways more straightforward than any of them, and it should be. We expect sort of a direct alcohol effect on FIB4. We had about 20% of values that were in sort of a concerning range over the cutoff of 1.45. It was positively associated with heavy drinking, and the odds went up with heavy drinking, so that's a positive odds ratio with heavy drinking, and then frequent heavy drinking also has a positive odds ratio relative to infrequent heavy drinking. So it was associated. Sorry, these are not odds ratios. I've done it both ways. These are actually just coefficients, but in odds ratios you get the same results, that being above that clinical cutoff is more likely with heavy drinking. There was no interaction, however, with HCV, with gender, or with antiretroviral medications. So we see sort of a non-complicated effect here, which we're a bit surprised by. We do have to take into account this sample did not have a high rate of HCV, so perhaps that could be explaining part of that. But it was positively associated with log viral load and also negatively associated with CD4, even when both of those were in the model together. So we do sort of see a general HIV-related health correlating with the FIB4 as well, and then alcohol being another layer on top of that. So when you try to think of like total effect, there's a lot to consider. That alcohol use is definitely changing adherence. Adherence may or may not be relating to viral load. Alcohol seems to be reducing CD4. It's worsening FIB4. If people are doing worse on their HIV outcomes, they're doing worse on their liver outcomes. So I would say that alcohol's total effect, when you think about the total picture, is maybe a little bit higher than it does when you look at each piece. For renal function, only 4% had sort of abnormal values on GFR. It wasn't significantly associated. That was not a big surprise based on the result that we had seen, so we didn't see that. But hemoglobin was a bit surprising. So the average hemoglobin was 14.5, 33% were below the level of 14, and 6.8% below a 12. And it was significantly and positively associated with heavy drinking. So this was going in the opposite direction. We sort of thought from previous data that that's possible, but I was a little surprised at sort of the robustness of that association, that the magnitude of that was relatively large compared to some of the associations we saw. It didn't seem to be a dose effect. So more frequent heavy drinking didn't seem to raise hemoglobin further, but engaging in some heavy drinking did. Now, people who drink heavily, at least 1 or 2 times a month, they also are drinking other days of the month as well. So, this could also be something if we divide it up as people who drink, a fair amount versus people who barely drink versus people who don't drink at all. Alcohol seems to have some effect on hemoglobin. It's not due to smoking status, so we did control for that. There was no indication it was that. It was independent of medication adherence. So when we look at sort of our total VACS score, given where this sample is, hemoglobin was more likely than most of our other things to be abnormal. So if anything, VACS scores would trend towards being better in drinkers because of hemoglobin. So we're sort of finding in this sort of this complex association where hemoglobin's doing something. Whether it associates with positive clinical outcome, we don't know. And I think that's really the question that we need to answer is, does the reference range need to be just changed for alcohol drinkers? And that was a question actually in a specific article actually asking for general population. But I think that's something we can look at, and as we look at the VACS data, perhaps we can go a little further with that. So lastly, we're trying to get at some causal inference in these data. It's difficult when you've got these wave data. So I've essentially just shown a repeated association over time. And all that does is kind of increase power, but doesn't necessarily tell us a lot about causality. We have a lot of potential confounds, depression, substance use, all of those things. And the concern is not that they affect alcohol per se, but that they also affect the association between alcohol and HIV status, and that they can have a direct effect on HIV status as well. So the inference can be strengthened if you try to do some sorts of causal modeling. And alcohol and adherence, in this you can think of them as being presumed to occur before the biomarker assessment. Now that's stretching the assumption a bit, right? So you're measuring the biomarker on that day, you're asking about the drinking from the 30 days before, but the biomarker obviously reflects stuff that's been happening during that time. But I think we can say that there's not a confound in that we don't think that the biomarker measured that day is what caused your behavior over the last 30 days. So I think at least there's enough temporal association to think that that's the way it has to go. Likewise, I think we can say that alcohol is more likely to cause non-adherence than the converse, that people are, they forget to take their medication, so they say, I should drink. It's probably less likely. I think what we're seeing is much more of the opposite. I plan to drink, so I'm changing. So there's some recent methods to handle these types of confounds with lag data. Valeria and Vandevelde have a really good paper in Psych Methods that goes into this and actually provides algorithms for SAS and SPSS to do it. So I would highly recommend that. Essentially, what you have is a model like this, and I'll walk over here a little bit just to point a little more easily. The key is that, so this is the outcome that we're interested in at time t, whatever that biomarker is. And then we want to control for what happened at T1. We also want to control for heavy drinking at T1. And then essentially all these other things are our potential confounds. So whether they're on medication or not, and then whether or not they're adherent. So you have to kind of create a way to deal with that missing data. And then from that has sort of the beta path that would be affecting our clinical outcome. We have drinking having its direct effect. And then we also have substance use as a confounder, depression and employment as potential confounders, kind of psychosocial changes associated with drinking as well. And then we can take all of these and we can lag them back one so that we have drinking at a current assessment and adherence at a current assessment. We can take a t-1 of all of those and control for that. And essentially what you're then modeling is, does change in drinking in a given follow-up assessment tell you anything about where they were from their previous assessment and their previous level of drinking and their previous level of depression, employment, et cetera. So it gives you sort of that more clean measure of change, and it also allows you to decompose effect into a natural direct effect and a natural indirect effect. And those are sort of like the sort of Barron and Kenny's indirect and direct effects, but there's some other stuff you can do in the counterfactual model of mediation that lets you say a little bit more about that. I won't bore people with too much of those details. But this is fairly preliminary right now, so I don't want to present numbers as if they are the final be-all, end-all. But what we see is natural indirect or mediated effects of heavy alcohol use on viral load and to a lesser extent CD4 through its impact on antiretroviral adherence. So what that's saying is as people change their drinking over time, their adherence changes at the same time point, and we do see this signal that seems to lead to changes in viral load. So you can have a non-significant direct effect, but you can show that the pathway does, in fact, go through adherence. We see no significant natural direct effect. And again, this is important to note that this is change, right? So this is how much you're changing your drinking. We're controlling for where they were drinking before and what their outcome was before. Their outcome before could be caused by a whole lifetime of heavy drinking, right? Your FIB4 value is caused by 10 years of heavy drinking, and we start measuring you at the end of those 10 years. This is whether or not you're changed. So this is important for patients to know, and this is important for us to know clinically. If we get change, yes, we expect that could impact medication adherence, and that could affect both of these outcomes. We also found a natural direct effect of heavy alcohol use on FIB4 and no significant natural indirect effect. And that's interesting, too. A lot of clinical trials show that changing heavy drinking does change AST and ALT. We're able to show that in sort of a very long-term observational type of study with a lot of observations. And that seems to be a reasonably robust effect, and that's not through adherence. So it suggests that, if we're thinking of the total picture, all three of these are good predictors of survival, that alcohol is having an effect on all of those, but it maybe is happening through different pathways. So hopefully we'll have those results finalized over the next few months. So this gives us sort of this is just sort of one more piece of information, like stacking it all on there. So we know that, yeah, alcohol's doing something, and we're learning a little bit more about how and why and where. And of course, we need to do something with it, right? So we need to have some types of interventions, and interventions hopefully that harness some of this information to enable clinicians to be better at what they do, and also patients to have information that they need. So there's been a number of clinical trials to try to reduce hazardous drinking in people living with HIV. Most of them have not targeted specifically people who are heavy drinkers and had a specific intervention just on alcohol use. So it's actually a relatively short list when you kind of take out that. The Parsons study was really, I think, the first that was specifically focused on heavy drinking. That was an 8-session MI plus CBT versus the same thing, but done in time and content equivalent kind of control. They found effects on all the biomarkers that we'd like to see and the behavioral markers, but not on alcohol use. So that was a little surprising. But it's important to note that both groups are getting the same information, and we assume that information causes a lot of change. And they did show a big change in alcohol use. So that's really just about the means of delivery. I think it's sort of imperfect. The Velasquez study is a little more clear. They had individual sessions plus an education referral only, which was really short. And this was, again, fairly intensive, though. They showed a 30% reduction in heavy drinking, an effect of sex risk as well for people who did combined alcohol and sex, but they didn't report biomedical outcomes, so we don't really know, are these effects going to filter down? But that sort of shows pretty decent effects that we're starting to go away by 12 months, but at least we're maintained somewhat. Becky Pappas, and working with colleagues here, has done a trial in Kenya to do CBT versus usual care in hazardous drinkers with HIV. And that one really is the first to show effects on everything. And that, in some ways, really should. They're in a resource-poor setting. They're giving an intensive intervention to one cell and really nothing to the other cell for alcohol. So that's sort of the ideal way to find out, is it likely to fall down? And in fact, yeah, they found the effects trickle down to adherence, to viral load, to CD4, to everything in that study. So I think that's kind of an exciting finding. This just sort of shows the drinking effects, and you can see that over time, the additional sessions really do bring down drinking, and it stays down in the experimental condition. But getting assessed makes you drink less too, as we know. But at least in this time, it came through nonetheless. Debbie Hassan has a study now, Impress and Addiction. I don't know if people know that it's an addiction yet, but that was HIV-infected population as well. They had sort of an advice condition, an MI condition, then MI plus a health call, and they've already published the pilot study on that, which is just interactive voice recording. What did you drink? Sort of every day. It gets people kind of continuing to focus on it, and they can get feedback on their drinking at the end of a month, what it looked like. So their MI is sort of enriched with the data that the people actually report. They found if you had non-dependent drinkers that all arms are doing a pretty good job of reducing drinking. No differences there, 50% reduction though. But that MI plus health call was particularly useful in dependent patients. So there does seem to be some sort of severity matching. So we're kind of getting there, I think. We're learning a little bit about the studies and maybe what might be important. But possibly, maybe a more minimal intervention is going to work. So in all that context, we are in the process of running a study now that's looking specifically at a brief intervention for HIV-infected MSM in an HIV primary care setting. The study is taking our work that we've already done in MI in the emergency department and also with smokers, combining it with work that I did with John Morgenstern on a goal choice intervention for alcohol use disorders in MSM who were not HIV infected. And in that study, what we found actually is if we had an MI condition versus CBT plus MI, and what this told us here was that MI actually made a quicker reduction in drinking compared to CBT plus MI, and then they sort of flattened out. And sort of that was consistent across a lot of variables we looked at. Our feeling actually in that was that MI has a certain advantage in being short and not saying we need to teach you anything, just being that you're ready to change and you can do it and here's the information. So that kind of encouraged us that maybe going fairly brief would be good and allowing people to have their own goals about is it abstinence or reduction as well was feasible. We've since done another trial. I think we're doing an analysis right now. I think there's only 8 that we found true RCTs in MSM for hazardous drinking. So this was only the 2 of those 8 here. So there's not a lot to go on when you're thinking about specifically tailoring interventions for MSM. We, in this case, had naltrexone and then a fairly involved MI and CBT-ish coping skills type of intervention versus people in the other condition got a really minimal sort of how you're doing on your medications, and that's about it. Really very, very minimal counseling. And what we found was that if you got naltrexone and that minimal thing, that's this top one, that you're drinking reduced but not that much. Okay? And then if you got naltrexone plus counseling or the counseling, that's the triangle there, you did pretty well. Maybe naltrexone acts a little faster than sort of skills training, and that makes sense that you would expect naltrexone should start doing its thing pretty quickly. But that it really didn't matter. Naltrexone wasn't adding much necessarily in that. So again, an indication that perhaps for a lot of people who are heavy drinkers but not heavily dependent, not being seen in addiction settings, that perhaps treatment without medication may be substantial enough. And I think sort of the trial going on here will be sort of another way to kind of tease that apart. So our study, and I'll just give you the very, very quick version of this. We're recruiting 224 HIV-infected MSM from Fenway Health. They're all hazardous drinkers. They can be dependent. They can sort of carry virtually any comorbidity with them that they would like except current IV drug use. And they can be depressed and sort of everything else. We're really trying to make it as much as we can like the clinic. They get treatment as usual, which is treatment as usual plus assessment, which is an intervention, or a brief intervention. Then we have follow-ups at 3, 6, and 12. Very briefly, they're drinking about 30 drinks a week, so this is quite a heavy drinking group. 34% of the days they drink heavily. Depressive symptoms are very high, all above the cutoff, and that's sort of typical in HIV populations. High-risk sex, they weren't screened for high-risk sex, so it's not necessarily high. So this is a secondary outcome in the study. Do we reduce high-risk sex? Antiretroviral adherence, only about 19% are not, sort of poorly adherent. Detectable viral loads, even lower than that. So again, the idea that not everyone has to be adherent to get there. CD4 counts only about 20% below 400. Abnormal liver function, however, we see a lot more. So this is sort of consistent with what we see that, our sense so far of the Sun data and this data that people can be in care and doing pretty well and drinking heavily, and even being non-adherent a fair amount. It won't necessarily show up in viral load or maybe even in CD4, but liver function may be one place where you're going to find it. HCV is very low in this group, so this liver function dysfunction is most likely from drinking, potentially from HIV as well. But given how well the viral load's controlled, my guess is that a lot of that would be the drinking. So we've done treatment development. We had sort of 5 participants. We went through a process of identifying the challenges and so on. The challenges were not only the severity of the psychosocial problems. That was a big part. People with a lot of mental health problems, psychosocial instability, residential instability, financial problems. Length of assessment and intervention, I'm sure we've all lived through that. Trying to make it shorter, but we're not getting there. And we needed it to be sustainable. We felt like one visit was just not enough in this case, even though we were thinking one visit with some phone calls, and so now we've sort of increased that a little bit. I'll tell you about that. And then we have the complexity of the feedback. And I'm sure I'm not going to get through everything. I won't go through MI. Anyone raise your hand if you don't know MI? Okay, so it's MI. They do all that. This is the other piece I wanted to mention, just as we're thinking about like data across studies, is we do the timeline follow back, and then we have to assess their drinking, their drug use, their sexual behavior, and their medication adherence, sort of on every day for 30 days. And this is sort of what it looks like. This is 1 day's worth of data. So this went through a lot of iterations, but essentially we're going to be collecting how many doses were you supposed to take, how many did you take actually, and if there was a difference, what were the reasons for non-adherence? So that was something we added based on Kalishman's work, and I'll show you a little bit of data on that. And then we also go through every single partner, anonymous, casual, first time, dating, long-term, male to female, et cetera, all the different sex acts that could have occurred, were they protected, et cetera. And there's still like 10 more questions we would like to ask, but it just goes on and on. So I think these kind of data are really rich and really valuable, but they take a lot of patient time and I assume a lot of coding time. But this was interesting. We found that we have a lot of days to report on from the first 75 people. 41% are having some missed dose in that 30 days. So it's actually pretty good adherence overall in the sample. We have 148 missed dose days, and 95 of those were drinking days. So definitely drinking is coinciding with the missed dose, although these people are drinking pretty high, like 50-something% of days. So there's just a base rate phenomenon there. But this was interesting. So we did look at the adherence, reasons for non-adherence, and this cuts off, but this is, were you drinking or not drinking on a given day? They listed a few more reasons on the day. This actually is higher than 148 because people listed multiple reasons when they were drinking and couldn't decide. Was it, basically, was it routine disruption on the drinking day, or was it unintentional lack of adherence? But this is the big thing here. This is substance use, so this could be other drugs too, right? We didn't feel like people could answer unless you made it more broad. But a lot of the non-adherence on drinking days that's substance-related, that the patient at least says is substance-related, they're saying are intentional. And they really are saying not that like while I was drinking I decided, but I knew going in that I was not taking my meds that day. It was a party day. So I think for intervention development, that's actually changing how we do our intervention. Instead of talking a lot about, well, when you drink, you can't control your intentions as well, and there may be all this impulsivity and stuff. In fact, we're really feeling that we actually have to go more at this and talk about sort of that intentional decision. The other things are, relatively more even across those. Sometimes psychological factors a little bit higher on those days, but not given the number of days we had. We had more, so that's not really an association. So not sort of statistical significance here, but clearly, clinically, we know we have to do our work in a way. So lastly, just to show you like what we do with an intervention. It's a lot like a lot of brief interventions would be. We have feedback. We do take this from the MACS study. We do, so this is normed for MSM in urban settings. So we give them a norm about their drinking level and where it fits in. And we find that for a lot of people, this is very impactful. You don't have to do it. If we're getting a lot of resistance, we don't go here because people then just get really upset. But if they're sort of open and engaged, then we kind of say, well, where do you fit in here? And all of our participants are fitting in here, and they also get a percentile. And this was sort of the feedback innovation, is we couldn't go on and on for pages. A lot of these people have cognitive impairments and so on. So we had to do it in one page of feedback. And what this does is based on our interview with them and their medical record, we type in a bunch of numbers, and then these arrows light up depending on the data that we put in, and then we print that out for this individual patient. So a red arrow for medication adherence would mean it's less than 90%. It would be yellow if it was less than 95%. Sex risk, they're engaging in some risky sex, but not necessarily high transmission risk sex. Brain dysfunction, in this case, this person has one subtest on the neurocognitive assessment that they had a deficit in. If they have two, then that goes to red. Liver health, again, that's pretty obvious. Substance use, sort of, again, we light that up for, depending on how many, whether it's multiple substances, how often it happens. Smoking, we sort of would always light that up as red. And then viral control, people are usually pretty good, but again, that can go depending on sort of your level of viral control and actually immune function. CD4 fits in there too. And then we have this sort of back page, and what this does is we take that timeline data that we've talked about and we try to use it in the clinical intervention. So right up here, for this person, it was days not adherent in the past 3 months. So that was based on ACASI. So we also have sort of a broader measure that's hopefully more impersonal. We talk about that, and then from the timeline follow back, we say you missed 4 days in the last 30. And on those, on the days you drank, you missed 3 times, and on the days you didn't drink, you only missed once. You know, what do you think about that? Why is that association there? And then we have the same thing for unprotected sex, too. And this really leads to a rich discussion where people can say, A, I don't like taking my pills on the same day. I think it's a problem. Or when I drink, I'm always at a friend's house and I'm never around my pills. All sorts of different things. Sexual health, we get very complex responses where people are saying, I'm making real decisions about what's high risk and what's not, and who I can be with, and who I use a condom with, and who I don't. And so sometimes it sounds very intentional, and alcohol is just sort of part of it. But they didn't violate any of their intentions. Other people really are saying that they really feel like they're losing control when they drink. And so that's really, really helpful and important to get that sense, and this data helps us do that. So lastly, and I'll just spend a minute on this so I have time for questions. Because these interventions are new and we won't have outcome data for a while, we're going to start kind of slugging through them and figuring out what is it in the intervention that works, what doesn't work, what counselor techniques are effective in getting people to consider change and commit to change. And so that's what this is related to. We've got co-investigators here in Hopkins and UAB. And essentially what we're trying to do is state-of-the-art mechanisms research on these alcohol HIV interventions to break down what happens in the session, and I'll tell you that quickly, and then to be as a national resource that we can help other studies that are doing this kind of work do this. So we have these participating trials, 2 of those you recognize from here, 2 I've mentioned, and then the one that's a CNIX trial going on at UAB. What we have is that everyone's different. We've got different providers, social workers, medical providers, paraprofessionals. Sometimes they're using MI explicitly, sometimes they're not. Sometimes alcohol is the target, other times it's more secondary. Sex risk is discussed, it's the primary outcome in this study. Adherence is discussed in some studies, it's the primary outcome in one study. Sometimes there's alcohol pharmacotherapy, sometimes there's not. So we have all sorts of different topics and contents in that. So we're trying to take the motivational interviewing skills code and map that on so we can figure out what happens. Are they using MI types of techniques? And if they're not, is that a problem? And then we have to code sort of every single utterance and what was the topic, what was it about, and how did they talk about the topic to determine if counselors, whoever they are, may be worse at certain kinds of discussions with certain kinds of patients under certain kinds of conditions. And quantifying what's worse and what's not is the trick, but we'll skip through that. But we're trying to figure out, does provider language predict behavior, client behavior, and does that down the road. But essentially everything, for just if they're talking about alcohol, you have to know, are they talking about the patterns, the consequences, barriers to change, facilitators, their goals, treatments they've tried, what they're doing in the meanwhile to kind of make a change in a bigger thing, what are they doing that kind of getting in the way of making a change. So you have to kind of code every single utterance. Is it something of that? And then within that, you have to decide what's the speech act. And this is a partial list. It goes on and on. I only gave you one little piece. But is that are they just asking for information? Are they giving advice? Are they confronting? Are they expressing an emotion, expressing empathy, making hypotheses, et cetera? So eventually we will have gazillions of data points and hopefully enough time to sort it all out. And so we got to transcribe about 1,000 tapes, and we're just starting to kind of apply these now. And then we'll be able to use those causal modeling methods to look at what are those sort of interactions mediationally as well. And so that's hopefully something, in a few years, maybe I'll be able to come back and tell you where we've got on that. And so on that, I'll say thanks.